Introduction. Welcome to today's lecture on the histology of the renal nephron and its role in renal function. In this comprehensive script, we will explore the intricate structure and organization of the nephron, the various mechanisms of transport involved in renal filtration, and the clinical implications of nephron dysfunction. By the end of this session, you will have a deep understanding of the histological aspects of the nephron and its vital role in maintaining homeostasis within the body. The nephron is the functional unit of the kidney, responsible for filtering and reabsorbing substances. It consists of the renal corpuscle, glomerulus and Bowman's capsule, and the renal tubule, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. This structure allows for regulated filtration, reabsorption, and secretion. The kidneys rely on a robust blood supply to function properly. Around 20% of the cardiac output reaches the kidneys through the renal artery, which branches into smaller arteries. These arteries supply blood to both the renal cortex and medulla. The afferent arterioles within the cortex give rise to the glomerular capillaries, forming the glomerulus. The blood is then drained by efferent arterioles into the paratubular capillaries, which surround the renal tubules. This arrangement facilitates efficient filtration and reabsorption processes. Ultimately, the intricate vascular network ensures the nephron receives vital nutrients and oxygen while eliminating waste products. The loop of Henle is an important part of the nephron that helps concentrate urine. It has three regions: thin descending limb, thin ascending limb, and thick ascending limb. Each region has specific functions and specialized cells. The ultrastructure of the loop of Henle facilitates urine concentration through countercurrent multiplication. In the renal tubules, transcellular transport mechanisms play a significant role in the reabsorption of various substances. These mechanisms involve the transport of molecules across the epithelial cells lining the tubules. For example, glucose is reabsorbed through sodium-dependent glucose transporters, while amino acids are transported through specific carrier proteins. Proximal tubules are particularly efficient in reabsorbing most of the filtered load of glucose, amino acids, and other solutes. Active and passive transport mechanisms within the tubular cells ensure the selective reabsorption of essential substances while allowing for waste excretion. Alongside transcellular transport, paracellular transport mechanisms contribute to the reabsorption of substances in the nephron. Paracellular transport refers to the movement of substances through the intercellular spaces between tubular cells. These spaces are regulated by tight junctions, which determine the permeability and selectivity of the paracellular pathway. Small ions, such as sodium, chloride, and potassium, can passively move through these spaces due to the movement of water and electrochemical gradients. Paracellular pathways allow for the fine-tuning of reabsorption, maintaining the balance between water and solute concentrations. The thin descending limb of the loop of Henle, lined with aquaporins, allows water to passively diffuse out of the tubular fluid into the interstitial space due to the high osmolarity in the renal medulla. This concentrates the fluid for reabsorption in the ascending limb, aiding in water homeostasis. In the video on the loop of Henle, we will discuss the role of the thick ascending limb in ion reabsorption. This section of the loop reabsorbs sodium, chloride, and potassium ions through active transport mechanisms, helping create a concentrated interstitial fluid for water reabsorption in the collecting duct. The loop of Henle plays a crucial role in the dilution and concentration of urine. By establishing a counterflow multiplication system, the nephron can create a hyperosmotic interstitial fluid in the medulla, aiding water reabsorption. The descending limb concentrates tubular fluid as water is reabsorbed, while the ascending limb dilutes fluid by reabsorbing solutes. These mechanisms allow the nephron to adjust urine concentration based on hydration levels. The counterflow multiplication process in the loop of Henle establishes an osmotic gradient for water reabsorption. As fluid descends in the descending limb, solutes are reabsorbed while water remains, leading to concentration. In the ascending limb, solutes are reabsorbed, maintaining the gradient and generating a hyperosmotic interstitium. The intricate network of blood vessels within the kidney is of utmost importance for proper nephron function. The combination of glomerular and paratubular capillaries ensures efficient filtration, reabsorption, and secretion processes. The high hydrostatic pressure in the glomerular capillaries facilitates the filtration of plasma into the renal tubules. Subsequent reabsorption of substances occurs in the paratubular capillaries, allowing for the reuptake of vital molecules into the bloodstream. This intricate vasculature ensures the maintenance of plasma composition and plays a crucial role in the regulation of body fluid volume and solute balance. Various factors regulate the function and activity of the loop of Henle. Hormones such as antidiuretic hormone, ADH, and aldosterone play essential roles in water reabsorption and sodium reabsorption, respectively. ADH promotes the insertion of aquaporins in the collecting duct, enhancing water reabsorption. Aldosterone acts on the distal convoluted tubule and collecting duct, increasing sodium reabsorption while promoting the excretion of potassium. Additionally, factors like blood pressure, glomerular filtration rate, and osmolarity of the tubular fluid all influence the function of the loop of Henle and overall renal activity. Understanding the histology and function of the nephron is of great importance in the diagnosis and management of renal diseases. Disturbances in the different segments of the nephron can lead to altered filtration, reabsorption, or secretion of various substances. Conditions such as renal tubular acidosis, diabetes mellitus, and polycystic kidney disease can result in impaired nephron function and compromise overall renal health. The ability to interpret histological changes in the nephron allows for accurate diagnosis, prognosis, and targeted treatment of these conditions. Understanding the histological structure and function of the loop of Henle is essential in comprehending the complex mechanisms of renal filtration and reabsorption. This knowledge is crucial for healthcare professionals in diagnosing and treating renal disorders effectively. It also highlights the importance of maintaining renal homeostasis for overall health.